lecture, we have a return from Professor John Rogers at Northwestern, and we'll introduce the panel after his talk. So Professor John Rogers is back for return engagement. He is the Lewis Simpson and Kimberly Query Professor of Materials Science and Engineering, Biomedical Engineering, and Medicine at Northwestern University, with additional appointments in mechanical engineering, electrical and computer engineering, and chemistry, where he's also the director of the Query Simpson Institute for Bioelectronics. He's a co-inventor on more than 100 patents, has co-founded several successful technology companies. Among his many awards are MacArthur Fellowship, the Lemelson MIT Prize, the National Security Science and Engineering Faculty Fellowship, the Smithsonian Award for American Ingenuity in the Physical Sciences, the MRS Medal, the Benjamin Franklin Medal from the Franklin Institute, and a Guggenheim Fellowship. He's on our editorial advisory board and a frequent author at ACS Nano. He's a member of the National Academy of Sciences, National Academy of Engineering, National Academy of Medicine, National Academy of Inventors, and the American Academy of Arts and Sciences. It's my great pleasure today to introduce my friend and inspiration, Professor John Rogers. John, the floor is yours.
Yeah, great. Thanks so much for that kind introduction, Paul. It's good to see you virtually. Hope all things are well at UCLA. Also, thanks to Alice for inviting me to give another lecture in this amazing series. I think Alice should be congratulated for all the efforts that she's put into this virtual seminar series. I think it's a tremendous contribution to the community, and almost at the level of Paul's role as chief editor at ACS Nano. You guys are doing amazing things in service to the scientific, broader community and society. So thank you guys for everything that you do. So I'm going to go ahead and share my screen and get started and tell you about some things that a little bit different than what I talked about when I participated in this seminar last year. Hopefully, kind of interest to the audience here. So let me go ahead and get started. Hopefully, you can see my screen at this point. Let me get to my first slide. So I put together a deck. I'll talk for maybe 50 minutes or so on functional nanomaterials for neural interfaces with a focus on 3D neural interfaces, which is sort of a frontier that I think is pretty interesting. And there's some basic material science involved, but also with a strong intersection to the biology community with potential relevance to how we think about medicine and treatment of disease as well. And so it's an interdisciplinary topic that we feel is pretty exciting. So we have some significant efforts in this space and been working in this area for some time. And so as Paul mentioned, my core expertise in my home department is material science and engineering. But as you'll see in this presentation, and as is reflected in many academic programs of research these days, it's very highly interdisciplinary, and you'll see that interdisciplinary flavor, I think, come through, in this presentation. But I sort of broke out the talk into four parts. The first one is fairly short, but I wanted to make a few statements of background and to try to motivate our interest in this area. I'll then talk about some of the foundational concepts that we've been working on over a number of years, actually, around how to create functional nanomaterials, to manipulate them, to integrate them into devices and ultimately full integrated systems. But in the context of this presentation, with a focus on neural interfaces. And I'll show you what's possible in terms of thin, flexible sheets that can integrate with surfaces of neural tissues, as well as filamentary probes with cellular scale device components that can be used to interrogate and manipulate tissues at depth as two complementary platforms where I feel like we and others who are doing work in related streams of research have been able to make good progress over the last 5-6 years or so. But then really kind of dive into where I think the frontier resides in terms of new concepts and maybe ultimate forms of biointegration, where we're attempting to distribute these functional nanomaterials and devices over three-dimensional volumetric spaces rather than on surfaces or at individual targeted locations at depth. And I'll describe a couple of approaches that we've been working on for creating those 3D architectures and then integrating them with living systems, and give you a sense of where we stand, and where there may be opportunities for new ideas and new materials approaches. So that's kind of the overview, and I'll go ahead and just dive right in. And so why should anyone be interested in functional nanomaterials for neural interfaces? And I think it's ultimately because of the potential for using successful outcomes and leveraging them to develop new schemes for treating human disease. So thinking about patients ultimately, and improving patient outcomes, and maybe developing a portfolio of approaches that could complement more traditional pharmacological schemes for treating disorders, and where you really focus on both the central and the peripheral nervous systems as points of integration, as control levers to manipulate and modulate and improve the health of vital organs ultimately. And I think this idea of sort of a bioelectronic form of medicine is not necessarily new. And if you take a look at the illustration and the list on the left hand side of this view graph, you'll see a number of examples where that kind of scheme has been successfully used to treat patients with various sorts of conditions. And maybe the most prominent and well appreciated scheme of that sort involves deep brain electrical stimulation to treat symptoms of Parkinson's disease. Actually, it can be much more effective than any kind of drug-based therapy. So it's basically, coarse arrays, small arrays of electrodes on penetrating probes interface to relevant region of the deep brain, whereby electrical stimulation can eliminate effects of Parkinson's as manifested in physical tremors. And it's amazing technology, FDA approved, used fairly widely, but that's not the only one. I mean, you think about retinal implants and cochlear implants to restore a sense of vision and hearing, using again sort of electrical interfaces to neural tissues, and interfaces to the spinal cord as well, to treat various forms of chronic and neuropathic pain, peripheral nerve interfaces also. So I think there is some amount of momentum around using these kinds of electrical interfaces to treat disease. But I think it's just the very earliest days of thinking about that type of scheme. And as material scientists and as academic researchers, you get asked, how can we contribute to improving the efficacy of these kinds of approaches, and maybe expanding the modalities for doing neuromodulation, and where would the opportunities be that could take us beyond just a small countable number of electrodes as an interface into new realms of control and reliability and chronic stability. And so I tried to identify, and we wrote a review article that's published in Cell last year to try to outline from the vantage of an engineer and a material scientist what topics might be interesting to contemplate in this area. And so you might think about modulation schemes that go beyond electrical, so optical, for example, and there are various schemes now that use genetic modifications to create light sensitivity and targeted classes of neurons that allow optical means for stimulation or even inhibition of neural activity with cell type specificity. And that would be much more powerful than just a blunt instrument based on electrical stimulation. But you might also consider, microfluidic delivery platforms that could allow for programmed pharmacology and drug delivery at targeted locations where you have tight temporal control over how those drugs are modulating neural activity. Newer approaches that the number of researchers are exploring, we are not, but I think it's an exciting area, where you use thermal modulation to control neural function, or even mechanical perturbations can have a pretty important effect. So thinking about going far beyond just electrical to these other forms of neuromodulation, and maybe mixing and matching them and using multimodal approaches for modulation would be interesting. And then, maybe combining sensing as well, measuring the biochemistry associated with neural activity, or, recording biopotentials or biophysical effects, and combining, modulation with sensing in some kind of closed feedback loop for very tight control, for augmenting the functions of key organs, and stabilizing their operation, and thinking about it, at least conceptually, in the way that pacemakers operate, where in many cases you measure the heart rate, and then you stimulate based on whether the heart rate drops below a certain threshold. And so it's sort of closed feedback loop, but doing that in the brain or across the peripheral nerves might be an exciting frontier to contemplate. So those are some kind of obvious areas that one might consider. And then, in terms of the underpinning technologies that would allow for those new approaches to modulation and sensing, what would you want to think about there? Well, ultra miniaturization would make a lot of sense. You just reduce the device load, on the body. And so making things as tiny as possible is an obvious kind of direction to contemplate. And that's where sort of functional nanomaterials come into the conversation. But not only miniaturized, but also, spread out in many cases over large areas where you need to engage, as I'll describe a little bit later, volumes of tissue. And how do you do that in a way that's biocompatible, not only from the standpoint of the materials, but also from the geometries and the mechanics. So thinking about soft, stretchable mechanics in functional semiconductor devices, complex architecture, not just penetrating pins and flexible sheets, but 3D networks, open networks that can engage at a qualitatively more intimate level with living tissues. And doing everything with wireless power and data transmission might be interesting. So there's some compelling opportunities there in electrical engineering primarily. But I mentioned closed loop control. And then as an additional thing to contemplate, I mean, many patient conditions are transient, maybe associated with a wound healing process. And so in certain circumstances, you don't need permanent devices, to address those needs. And in fact, a permanent device may have key disadvantages because, it would require a secondary surgical extraction after the device is no longer needed. So thinking about bioresorbable materials and devices in this broader context is another direction that one can consider. So anyway, I just want to paint the picture. There's a broad landscape here, many outstanding groups working in these directions. There's a sort of a compelling opportunity to impact human health, but where I think ultimately material science and engineering science more generally is going to be a key driver in allowing for progress in this kind of area. And so, ultimately, you think about, the human brain as biology's most sophisticated form of electronics. You'd like to bring to bear to that organ system, as well as other parts of the nervous system, man's most sophisticated form of electronics in one way or another. And so your attention would then naturally turn to the most advanced forms of silicon integrated circuits. And that's a spectacularly sophisticated form of technology, but it's intrinsically incompatible with soft neural tissues. These technologies, as everyone knows, are perfectly planar in their geometry. They're mechanically rigid. In many cases, they involve all sorts of materials that are not compatible with a biological environment. In particular, warm salt water essentially is the way you need to think about it. So most of these classes of materials will corrode when exposed to that kind of environment. And so it does provide some kind of aspirational goal to kind of think about silicon integrated circuits in this broader context, but it really requires a radical reconfiguration and reformulation of a wafer-based type of electronic or microsystems technology to allow for the kinds of neural interfaces that I was referring to in the previous slide. But it gives you sort of a goalpost to think about in terms of sophistication in function, and really strikingly contrasting with the kinds of very primitive, just passive metal electrode arrays that are used currently in the FDA approved devices I mentioned previously, just a small number of electrodes, and really limiting the level of function and the type of engagement that's possible. So going from just a few electrodes to maybe a few tens of millions of electrodes might be open up pretty interesting opportunities for treating disease. And the question is, like, how do you do that? And maybe how do you do that in a way that doesn't abandon all of the sophistication that's emerged from the consumer electronics gadgetry industry in terms of computational capabilities and control and so on, enabled by extreme scaled silicon CMOS. So that's kind of the idea, and that's a topic that we've been thinking about for a long time. I guess initially in the context of electronic drivers for flexible displays way back in the day when I was at Bell Labs and others as well, obviously. But like, how do you build flexible electronics? And then, over the years, how do you build biocompatible, stretchable electronics? And then how do you think about neural interfaces? So this is kind of going to go way back in time. And I don't want to spend much of my talk on these topics, but just to set the context, like how do you build a soft, stretchable, or at least flexible piece of high quality electronics? One way to do it would be to use carbon nanotubes as a functional semiconductor material, or maybe use polymer based semiconductors and you put them on plastic and you build devices in that way. And we spent a lot of time working in those directions, and a number of very outstanding groups kind of thinking about that. But then the other way would be to say, well, how can I maybe reformulate the way that silicon is used so that I can maintain some degree of alignment with the broader semiconductor industry and move forward in that way. And so that kind of scheme has worked better for us, at least, than some of these other concepts. Not to say that they don't have promise, but just in our hands, these kinds of inorganic hybrid systems have worked out pretty well, I would say, by many measures. And so the concept here is not necessarily to reinvent the way that silicon is grown and purified and doped, but to take what is a very advanced piece of materials technology, in the form of a silicon wafer at commodity cost, and then slice it up into very tiny pieces that are thin enough to be flexible and small enough to allow kind of cellular scale integration with neural tissues. And there are a variety of ways to do that. This scheme is one that has worked pretty well for us. We use anisotropic etching techniques to slice off the near surface region of a wafer, very thin ribbons of silicon, thin enough, kind of in the 150 nanometer range to support all the kinds of electronic functionality you ultimately need, but thin enough to be mechanically flexible so that they could be used as building blocks, as active semiconductor materials for devices you could build on a sheet of plastic or a slab of an elastomer, for example. And you can work your way down through the thickness of a wafer in a serial sequential set of etching processes, or you can do it in one shot by sculpting the sidewalls of these etched trenches that you develop in the surface of the wafer. These are wafers where you use TEMA or KOH to anisotropically shave these layers off the near surface, and you can do that in multi-layer stacks. Just in terms of throughput, you can create really mass quantities of these materials with semiconductor device grade properties, because they're being derived from wafer-based sources using the most well-developed crystal growth techniques. So you can do that. You can also do similar types of things with 3.5. So silicon's great for transistors, not so great for optoelectronics. And as I'll describe in a little bit, light emission and light detection turns out to be really important in the context of these optical approaches for neuromodulation and sensing. And so you really want to be able to mix and match, say, silicon with gallium arsenide or indium gallium nitride. And how do you create, functional nanomaterials out of those classes of semiconductors? And a variety of ways to do that. In this case, we're using a multi-layer epitaxial liftoff scheme where we grow using MOCVD techniques, alternating layers of aluminum arsenide and gallium arsenide. And then you etch down through those stacks vertically to expose the sidewalls. And in this case, you use concentrated hydrofluoric acid to eliminate the aluminum arsenide without etching the gallium arsenide. There's an etching rate contrast of about a factor of 1,000,000. So gallium arsenide then is lifted off the source wafer in bulk quantities in a similar kind of multi-layer scheme, much different, in the details compared to what I showed in the context of silicon in the previous slide, but conceptually similar. You're using a wafer as the basis, and in this case, you're just using the wafer catalytically in a sense, because you can reuse it for additional cycles of epitaxial growth. So you can do all of this. There are many other kinds of schemes and chemistries that can be used with other classes of semiconductor crystalline materials to create these functional nanostructures. But then the next question is like, how do you move them around, and how do you integrate them into systems in a deterministic way? And there, we've kind of adapted techniques of soft lithography, which I got involved in, back when I was a postdoc with George Whitesides. In those days, we were sort of printing molecules, but you can also print nanostructures, and so you use the wafers as inking pads and you print, and there's all kinds of schemes in soft interface physics that can be exploited to modulate the degree of adhesion of these nanostructures to the surfaces of elastomeric stamps, and you can move things around in this way. And Paul and his group have done some really nice work in printing solid materials, as well as a number of other groups also. So this turns out to work really well. I'm not going to describe the details. It's sort of outside of the scope of this presentation, but it works extremely well, really at industrial scales, and there are automated tools for doing this kind of printing installed at companies around the globe. And so the throughputs millions of elements per hour, and extremely tight control and overlay registration and so on. And these are some printing examples we did in our labs back at UIUC some years ago, just to show you how that works. So very, very tiny, thin semiconductor elements. You put them on plastic. There are certain aspects in interface, adhesion physics that lead to strong bonding because of the small thicknesses, and so you can bend these types of hybrid integrated organic inorganic systems without fracturing the inorganic materials or leading to their delamination. And in these kinds of arrays, now that's a great starting point for building functional devices. And I'm just going to fast forward. So that's kind of the base ideas in materials and manufacturing and mechanics. What do you do with all of that? And so we've been working in that direction for a long time, and others as well. But once you have a high quality semiconductor on a piece of plastic, you can do the rest of the processing pretty easily. So, it's easy to deposit metals and dielectrics at low enough temperatures that you don't damage the plastic substrate. You can do the photolithography and patterning and so on. And so you can build devices that are at practically meaningful scales that can be used as research tools and maybe precursors to devices that ultimately could be used for purposes in human health. And this is kind of the latest high water mark for us, based on our work in those kinds of directions. And so it's 1,008 channels. So what we have here is an array of electrode pads that interface to the surface of the brain. And then with a backplane, an integrated backplane of electronics, silicon nanoribbon-based electronics, which provides a per-channel amplification capability and affords an active matrix addressing scheme. So we only need lines to rows and columns. We don't need separate addressing lines to every unit cell in this array. And that kind of architecture, that active matrix architecture, is extremely important for any kind of scalable array-based system. So, a high-resolution display will use this same kind of architecture. So if you're thinking in the future of, tens of millions or hundreds of millions of sensing or stimulating nodes across a flexible neural interface platform, you really need to think about active matrix schemes, because passive addressing won't scale effectively, because the routing becomes impossible in managing the wires. You only need square root of n wires with active matrix addressing, whereas you need at least in wires without that. So it's an important feature. Probably critically enabling for 1,000 channels, but maybe one could consider at this scale some kind of passive addressing array, but not at a million. You'd really need the active transistor level functionality at a per channel basis, and that's what we've done. And some other things here that are important is like, how do you encapsulate these devices to keep the biofluids from corroding and shorting out the active backplane of electron, and many other aspects. And if you're interested, you can take a look at that paper. But just to give you a sense of how these basic materials and manufacturing and mechanical engineering ideas kind of come together to yield the neural interface of this type. And because the substrate is thin, because the functional materials are in the form of nanostructures, these nano ribbons of silicon, the entire system is highly flexible. Because the bending induced strains in these platforms are scaling down linearly with the thickness. And then we also use concepts in neutral mechanical plane design to further reduce the strains in the active materials to avoid fracture or damage when these devices are bent and twisted and conformed, to time dynamic curvilinear surfaces that you encounter with the brain or with the peripheral nerves or the spinal cord. So anyway, that's what it is. And if you look at the gain uniformity, you can do very well. Again, I think it's primarily because we're starting with excellent, materials properties derived from wafers. It allows you to do things like this, and low noise across the array as well. And so we work with Bijan Pasaran at NYU and have tested these devices in non-human primates. This turns out to be extremely difficult. A lot of practical things you have to solve in terms of the interconnects and so on, but you can do this. And this is 1,000 channels mapped out across the visual cortex. And what we're doing is we're creating a visually evoked response by presenting various images to the animals, and then we can map that out at unprecedented levels of resolution using these hybrid flexible electronic systems. And so this is kind of what the data look like, is tremendous amounts of data. And then kind of the next order question is like, what do you do with all this data? How do you interpret it? And how do you store it and transmit it and manipulate it and so on? So I think there are a lot of questions that could be examined in research efforts around data analytics and wireless communication. It's not so much our emphasis, but I just want to sort of highlight the fact that each one of these channels is being sampled at kilohertz sampling rates. And then you have 1,000 channels here, but into the future might have 1 million. So how do you deal with all the data, and how do you extract insight and meaning, and maybe how do you define patterns of stimulation, for example, that might eliminate electrical activity that's serving as the cause for an epileptic seizure, just as an example of how this might intersect with a topic in human health. So that's kind of how it works. It's useful to think about how it benchmarks against other approaches. And the key conceptual difference here is we have active matrix, as I mentioned before, and we have per channel amplification to drive the noise down that's otherwise dominated by Johnson noise in the interconnect traces. And so that's why there's kind of a separation here in this kind of plot: number of electrodes, density of sites. All the other reports focus on passive addressing schemes without the active semiconductor functionality distributed across the array. So that's the qualitative distinction, and there's a quantitative differentiation then that falls from that difference in overall architecture. So I think this is a direction where semiconductor nanomaterials will be enabling into the future high density mapping arrays. And so we've done 1,000 channels. I think for an academic lab to go to 1 million, it becomes pretty daunting, just in terms of what you can do with grad students and postdocs and academic clean room facilities. But we'd also like to conceptually think about what it would look like and how scaling might occur. And so, as I mentioned before, in any of these kinds of approaches, you'd like to be able to maintain some level of alignment between what you're doing and what the semiconductor industry is doing, because then you can leverage all the advances, the amazing advances in lithography and materials that are occurring in the consumer electronics space for these neural interfaces. And so this is an example of our attempt at that, where we did foundry sourced silicon CMOS on an SOI wafer. So here we're eliminating the buried oxide to lift off the surface region of the wafer, the active electronics in very thin, tiny forms, and then using printing to move those functional elements around. So it's not at the base material level anymore. We're printing functional, micro scale integrated circuits essentially. And so now we have per channel capabilities that can include data filter, digital filtering, and signal manipulation, and even data storage locally on a per channel basis. And with these kinds of soft lithography inspired printing schemes, we can spread that functionality out at low areal coverage, which is ultimately kind of what you need for these types of systems. And you can do that at the level of, I think this is 35,000 channels. So it's not interconnected into a functional system that we've tested on animal models, but at the base materials and manufacturing, mechanical engineering level, you can do all of this. And you can distribute the arrays in a way that anatomically matches areas of interest is another thing that we were demonstrating here. That's a natural consequence of this kind of soft lithography approach for manipulating these tiny functional elements. So this is just kind of our vision of how things might play out into the future as you increase densities and overall areal sizes. This is human brain scale size, as you can see here. So that's a few comments on what you can do in terms of scaled electrical interfaces. As I mentioned at the beginning of the talk, you'd like to think about other modalities of engagement with living neural tissues beyond just electrical, because electrical is great in the sense there's a long history of this, but it's non-specific. It's not cell type specific. So when you stimulate, you basically activate or inhibit neural activity across an entire volume of tissue that's in proximity to the electrodes themselves, which is not ideal, because you'd like to be able to pick out specific neural networks and just selectively stimulate or inhibit those networks, and you really can't do that with electrical neuromodulation approaches. The other thing is you end up with sort of parasitics associated with Joule heating. That can occur as you create eddy currents and so on. There are other kinds of disadvantages with electrical. The other thing is you tend to develop fibrotic capsules on these electrodes, and so the electrical impedance varies over time. And so you have to have some kind of scheme for controlling the voltage and the current amplitudes, because the thresholds vary as a result of the tissue growth that occurs across the electrodes. So a lot of challenges there, but it's still a direction that one should continue to consider, because there is such a long history of understanding around electrical. But I think the sort of newest developments, and this is occurring over the last 10 years or so, involve genetic modifications using viral vectors, for example, to create light sensitive ion channels in targeted neurons, so that illumination with photons can cause those channels to open up, or cause pumps to be activated in a way that allows you to excite or inhibit the activity of specific neuron types. And that's the techniques of optogenetics. And I think it's fairly recent, actually, that the kind of the material science and engineering community has gotten involved in this space. And Shing at Tsinghua has done some great work in this area, and it's really addressing the question of how do you introduce light into neural tissues, and sort of the biology, the neuroscience community was solving that challenge by just adapting fiber optic technology, from the telecommunications industry to create a conduit for injecting photons generated from an external light source down into neural tissue, as illustrated here in the depth of the brain. And that can kind of work at a certain level, but there are clear disadvantages associated with that type of scheme. The most prominent of which is that the animals are physically tethered then to that external light source, thereby modifying their natural behaviors. And actually preventing important neuroscience studies that might involve social interactions, because a pair of mice with these kinds of head stages and fiber optic cables in a single enclosure immediately get entangled with one another. And so I guess the vision from the engineering and the material science community, our group included, was that maybe you can get rid of the fiber optic cable and use some of these ideas in functional nanomaterials to embed the light sources themselves directly into regions of interest in the brain. And so that kind of launched us off in a direction of developing sort of cellular scale light sources and photodetectors that could be placed on filamentary polymer substrates to allow their delivery, these optoelectronic systems, to be delivered to regions of interest in the depth of the brain, or on the surfaces of peripheral nerves, or the spinal cord or the bladder or other places. Basically, shrinking the light sources down to dimensions that are comparable to individual neurons. And it turns out you can do that. It's basically using the same types of schemes that I've described up to this point in the context of silicon, gallium arsenide, and even silicon integrated circuits, the same type of material schemes, but now applied to indium gallium nitride blue LEDs. And so you end up with these extremely small, I think of the world's smallest LEDs. You can actually go much smaller than this if you want, but down to cellular scales, probably good enough, but extremely thin, very small, and capable of mounting on these thin polymer flexible filaments for injection down into the depth of the brain. And so you end up with a platform that's actually much smaller, much thinner, much more flexible than a fiber optic cable. And now you don't need the external tether, because your light source is embedded in your probe itself. And you can do multifunctional devices as well, LEDs and photodetectors and electrodes and temperature sensors and so on. And these filaments tend to be so thin that they're not even mechanically rigid enough to penetrate into the brain tissue. So you kind of have to use like releasable needles or some other vehicle to deliver these devices down into the tissue. And we use kind of a thicker polymer base and a bioresorbable adhesive to allow for that delivery to occur, but without increasing the sort of volumetric load of device technology into the brain, because the microneedle, the injection microneedle, can be removed. So that's kind of what it looks like. And we spent a lot of time in this area. It was the first paper is 2013, but we're continuously improving. And I'll give you an idea of sort of some of the things that we've done. And Xing's group has done some great work in this space as well. So this is kind of what it looks like. So these are 4 of these cellular scale blue LEDs, and mounted on a very thin polymer filament, and in this case threaded through the eye of a needle and wrapped around the shaft, just to give you a sense of the size scales that we're talking about. And so you can implant that down, you remove the injection microneedle. We have a wireless power harvesting and control unit that's built using the concepts of flexible, stretchable hybrid electronics that mounts subdermally just outside of the intracranial space. And tethers or actually electrically connects to the optoelectronic system that's implanted into the deep brain. And so you end up with a full system that's completely implanted, fully implanted, fully wireless, and can be controlled in real time. So you can turn on and off light sources. You can capture data from photodetectors, as I'll describe in a little bit, for monitoring neural activity. But the mice, and these are small animals, obviously, the mice that have these implants look and behave exactly the same as mice without the implants. And so now you can begin to look at these social interactions. You can look at behaviors of animals in naturalistic sort of 3D environments, all sorts of things that are not possible with the fiber optic cable. And you also end up with much better biocompatibility from the standpoint of the size. It's much smaller than the fiber optic cables, and the constituent materials are much softer mechanically, so more well matched to the soft properties of the tissues of the brain by comparison to a glass cylindrical shaft, that forms the optical fiber. So you can do all of this, and we've experimented and demonstrated that you can reproduce all of the kinds of optogenetic experiments that have been done with fiber optic cables, but now using these fully implanted wireless devices with cellular scale optoelectronics. So here we're using light to stimulate neurons that are responsible for the production of dopamine. It's a neurotransmitter that creates sort of an overall pleasure response in the animal. And so as a result of that, you can train behaviors without using physical rewards by just modulating the light up and down. And so here we've trained a place preference for an animal moving in this kind of Y maze. We activate the LEDs every time the animal is in this lower right arm of this Y maze, and over time they develop an affinity for that location of their environment, just to show that this can kind of work in that way. But I think the most exciting frontiers are in thinking about the neuroscience of not just the individual, but of social groups. If you think about humans, we're social animals, and you like to be able to understand brain activity in the context of social interactions, and that's what's kind of been being illustrated here. We have a place preference box where the left hand side of it is activated. So whenever the animals are in the left side of this environment, their optogenetic LEDs automatically turn on, and so they can go back and forth. They can interact with one another, and it really opens up some pretty interesting opportunities in neuroscience research. And I won't go into the details. This was just published a few weeks ago, but we've worked with neuroscientists here at Northwestern to really begin to leverage those capabilities in studying of social interactions. And this is a pretty wild experiment. So, and I won't get into details, just give you a high level description. So what we have here are three animals co-located in a single environment, and each one of them has a separate digitally programmable wireless optoelectronic system, and they're all optogenetically modified. So these animals, you can stimulate dopamine release independently in each one of them. You can also basically modulate prefrontal cortex behavior at different frequencies. And so what you can do then is you can turn on and off these different brain processes dependent on when the animals are in proximity with one another. So for example, animal A1 and A2 happen to be close to one another here. And so we have vision systems that pick up that local proximity. And whenever A1 and A2 are close to one another, we light up the cellular scale LEDs. It creates a pleasure response in the animals. And whenever A2 and A3 are next to one another, we do not activate, or A1 and A3, we do not activate, just when A1 and A2 are together. And over time, what we find is we can create social bonds. We develop sort of a natural infinity of animal A1 and A2. They develop a very strong bond. They do not bond with A3 because we're not optogenetically conditioning them to establish that bond. And then what's even more interesting is we can destroy that bond over time. We can recreate a different bond between A2 and A3. So it's kind of crazy type of thing, but it's essentially like friendship on demand. So anyway, this is kind of studies that you can begin to think about, and all the details are in that Nature Neuroscience paper if you want to see it. But fundamentally, again, kind of enabled by material science and engineering science, and collaborations with neuroscience scientists then to kind of leverage these new capabilities in fundamental studies of social engagements is one thing that you can do. The other thing is you can place these light sources really wherever you want. You're not constrained to the brain, where fiber optics, it can work because you have kind of a rigid skeletal structure to mechanically affix your fiber ferrule to the surface of the skull. But if you want to do a fiber in a soft tissue location and engage with the sciatic nerve, that becomes much harder. But with these soft cellular scale wireless LEDs, you just put them wherever you want them, and they work quite nicely. So this is sciatic, and this is spinal cord, and there doesn't seem to be any impediment to locomotor behavior with these devices because they're being built with these concepts in flexible and stretchable hybrid electronics, where all the mechanics and the form factors are optimized for biocompatibility. And you can see that animal running on a wheel. You can also do kind of other sorts of animal models. So this is an example of a study of birds. And so fiber optic cables are never going to work in this context, but with these wireless devices, they're small enough. You can begin to study unusual animal species such as birds, bats, snakes, fish, pretty much anything you can think about. You can put LEDs, optoelectronic systems in animals now. So basically introducing light is important. You can do certain things, neuromodulation, but in many cases, you think about closed feedback loop control, you'd like to be able to measure neural activity as well. And there are different ways to do that. There are genetically encoded calcium indicators, for example, whose fluorescence efficiency is modulated by changes in calcium concentration as a surrogate for monitoring overall neural activity. And so there you'd like to think about like a primitive, kind of spectrometer, actually, that would allow you to measure, that fluorescence efficiency as a function of time to sort of track neural activity. And it turns out you can do that, and I won't go through the details. It's basically combining these microscale or cellular scale LEDs with cellular scale compound semiconductor photodetectors and co-integrating them on these flexible filamentary probes. Will allow you to do that. So you can measure neural activity, you can stimulate or inhibit neural activity, and you can do all of that in a closed loop control manner with a single probe. It's kind of what you do. And so all the details here, is sort of beyond the scope of this presentation, but that's sort of one direction that you can consider. And this is just single wavelength measurements, but we can do optogenetic stimulation and calcium detection simultaneously. The back-end electronics start to get a little bit more complex here because now we have to have a data channel. It's not just modulating a device off, and on. You actually have to stream data back out to get the reading from the photodetector. But all that can be done in highly miniaturized forms. And again, battery free. I guess maybe I didn't fully emphasize that, but for these mice, a battery, even a tiny battery, is a massive, heavy object that can impede natural behaviors and also limit the operating life of the devices. So we'd like to operate without batteries. We're using wireless power transfer as a scheme for powering these devices, kind of the way to go. But this is what it looks like in an animal, again, fully implanted, and we're able to measure calcium and concentrations and fluctuations as a measure of neural activity at that local location in the brain. So these are the different types of kind of capabilities that begin to come together. And what I'd like to do in the remaining part of my talk, I think maybe 10 minutes or so, is to talk about where things might go into the future, sort of conceptually distinct from these sheets and these penetrating probes, but still leveraging the same ideas in functional nanomaterials, but now distributing those materials and device elements across a volumetric space, kind of in an open network geometry that may resemble the kinds of architectures that are fairly ubiquitous in biology. So you think about neural circuits. If you're sitting on the surface of the brain, you're really only interrogating the top 100 microns of tissue and most, or a penetrating probe, you're just kind of locally engaging tissue at the position of that probe. But there's a lot happening throughout the volume. It's an inherently 3D space, which is trivial observation, but it's important to keep that in mind. And it's 3D in the sense of an open network architecture. And you see that over and over again in biology. Vasculature structures, have that feature. Even at the cellular length scales, cytoskeleton, for example. And so, like, how do you, from an engineering standpoint, begin to design electronic and optoelectronic systems that include that kind of open mesoscale type network architecture is something that we think is worth thinking about. And you might immediately, turn your attention to 3D printing. A lot of great work in 3D printing, but if you look at what's done, it's typically plastics, or there's a lot of advances in metals these days. You can use plastics as sacrificial scaffolds for doing ceramics, different things, but you're not doing silicon CMOS. You're not doing device grade semiconductors. So it's not really. It may have a role, but not really fully satisfying. Kind of the vision you'd like to have for 3D and high quality semiconductor devices. There's some other kinds of shape morphing things that people have explored. Very very interesting concepts, but none of them seem to really fit what we had in mind in terms of 3D biointegration. And so we begin to think about how you could take maybe a planar wafer, semiconductor wafer, and somehow make it move up out into a three-dimensional space in some type of 3D architecture. And that seemed like it was going to be really hard, but it turned out to be really easy. And I'll describe it very, very quickly here. And the idea is that you use these approaches in shaving off the near surface region of a wafer-based platform. And that could be just at the material level, or it could be completed device structure. You shave it off the near surface to create what in this particular example is kind of an open filamentary mesh type structure. And then you move that mesh to a pre-stretched elastomer substrate. And you've configured the mesh such that when it comes into contact with that pre-strained elastomer, it bonds selectively at certain locations. And then you release the pre-strain in the elastomer. What happens is, through a buckling instability, you cause the material elements to move up out of the plane of the elastomer, the surface of the elastomer, in a coordinated collection of rotational and translational motions, which for this particular example transforms that planar serpentine mesh into a hierarchical structure of helical coils. Where you have one array of helices running along sort of the horizontal direction, and then another set of helices running on top of that first one, oriented in an orthogonal array. And you can do that with silicon, you can do it with gallium arsenide, you can do it with fully formed systems. This is an example showing that concept, and you can see the bonding pads here locally, and you can see this in cross section. And so these are the kinds of ideas, like really really simple sort of mechanical assembly schemes that allow you to go from planar to 3D in any kind of planar material system, any kind of planar device, multi-layer assemblies, pretty much anything. It's sort of agnostic to that. You sort of understand all the details of the mechanics, but this works quite nicely, and you can do this over large areas. It's parallel in the assembly. You can do not only single or double layer, you can do nested, space filling, entangled structures, all kinds of different things. I won't go into the details, but those are the ideas and the concepts that you can think about, as one promising route to this notion of 3D mesoscale networks with active functional nanomaterials. It doesn't have to be just filaments. You can combine membranes with filaments, and it can be multiple levels of three-dimensionality, and sort of the extent of 3D can be quite significant. Like the heights of these structures can be much larger than their base widths. For example, this is kind of a sunflower type structure that grows up out of the surface of the substrate using these kinds of buckling ideas. So lots of different things are possible, and this can apply to many different classes of materials, as I pointed out previously, and fully aligned, with microsystems manufacturing approaches that have been developed over the last 50 years in industry, which is important. So now you have these architectures. You can embed electronics and electrodes and LEDs, all different things. Then the question becomes, like, how do you integrate a 3D framework like that with biology? And I guess it's probably not going to work to take the fully formed 3D and then push it into a tissue system, because the amount of damage that you would impart would be quite significant, much more than just a penetrating probe. So you maybe have to think about it in different ways. So one way is to grow tissues around these structures. So now the structures are acting as kind of skeletal frameworks for growth of neural tissues, and maybe you use this as an in vitro model for neural studies, right? Neuroscience studies. And we did that. And so you can seed cells on these frameworks and grow them in dense colonies across these kinds of systems, and you can probe individual firing activities. You can stimulate different things as sort of a basket structure. It's just an array of electrodes, but interfaced with neural networks that are forming naturally in 3D. So going beyond what's traditionally done in two-dimensional multi-electrode arrays into things that are more reminiscent of actual biological structures in terms of the three dimensionality. So now growing it into 3D. So that's one direction that you can consider. I guess the area that we've spent more time on actually is to think about how to use 3D to integrate with organoids, spheroids, and assembloids. And these are for people in the audience who are unfamiliar with this. These are sort of relatively new tissue culturing techniques that allow for the growth of organ-like structures, but in miniature. So millimeter scale brains, for example, like mini brains that can be grown from human stem cells. So the advantage here is you can study neural processes in brain-like structures with patient-specific genetics because they can be grown from that patient's own cells. Stem cells. And so these are techniques that the biology community has developed, pretty interesting, but you end up with not 2D, neural constructs, but 3D. And then the question is, how do you probe them? You can't really probe them effectively with a 2D multi-electrode array because you have a 3D structure trying to interface with a 2D array. It just doesn't work. Maybe you can measure just at the base of that mini brain, but you'd like to get full 3D coverage. And so that's a direction that we spent a lot of time on, and appears to be promising for us. So the question is, how can you create like a 3D basket of very flexible functional elements that could gently wrap around an organoid to allow that kind of 3D integration. And so you can do all of that using these same kind of mechanical buckling approaches. And so we published this maybe a month or two ago. And so it's starting with a 2D active system and then buckling it up, in this case designing it so that it forms this enclosing kind of cage or basket structure, shape matched to the size and the dimensions of the brain organoid or spheroid of interest. And so here we have all kinds of different sensors and actuators, microscale LEDs, electrodes for stimulation and sensing. We have thermal actuators and temperature sensors, electrochemical sensors as well. So you can do lots of different things, again, because you're well aligned with planar device processing, but all the three dimensionality happens as a final sort of geometric transformation process at the very end. And so because you're doing this 3D assembly on an elastomer substrate and all the mechanics is elastic, it's reversible. So you can create the basket structure, then you can open it up by locally stretching the elastomer substrate so that you can gently insert one of these brain spheroids into that basket and then just gently enclose the functional elements around the organoid afterward. And that's what's illustrated in this finite element modeling movie. So we've opened up the basket, we've inserted the brain organoid, and then we've enclosed the organoid with the functional elements by releasing the pre-strain in the elastomer. And you kind of see that in the bottom sequence of images here. So you have the organoid, and it's becoming enclosed, and we do everything at a quantitative level with mechanics modeling through collaboration, long-term collaboration we've had with Young-Gang Huang here at Northwestern. Very, very important part of this project, all the mechanics theory that goes behind this. But now you can do 3D, so now you can look at electrical activity around the entire spherical surfaces of these brain spheroids derived from human stem cells in this case, and you can kind of see how that works. And you can begin to ask questions of what kinds of fundamental biology questions could you begin to ask with these systems. And so you don't have to have just a simple basket structure. We have all the theoretical modeling. We know how to design the 2D precursor and how to design the bonding sites and the pre-strain to create more elaborated interfaces to these spheroids. And in this case, we have a pair of spheroids. enclosed in a structure of electrodes that's forming an architecture that holds those spheroids into proximity with one another. And eventually, over a period of days, a neurite bridge will form between them. And so now we can probe not only just through optical microscopy when that bridge is forming, but when it becomes electrically active. And when the firing activity in A becomes correlated to that in B, indicating sort of a communication channel opening up between these spheroids. And you can watch all of that happen, in real time. And then as a next step, you can look at processes of not only formation of that bridge, but neuroregeneration of the bridge after it's severed. So you can come in with a scalpel, you cut the bridge, and then you can watch it reform, not just with microscopy, but now with electrophysiology to look at when that bridge becomes electrically active. And so, again, sort of platform for studying basic fundamental processes, in this case, in neuroregeneration. And you don't need to stop there. You can develop frameworks for creating assembloids of 3 of these spherical cortical spheroids and other things, and you can make arrays of these kinds of 3D structures for getting good statistics around studies of this type and so on. So in the last 5 minutes or so, I'm not sure exactly how much time I have, but I'll wrap up in the next 5 minutes. I'll show you some unpublished work where we're thinking about, especially in the context of this tri-spheroid assembloid, what's going to limit, like, the assembly of these different building blocks. So you can do neuromuscular junctions with, neural spheroid and a muscle tissue spheroid, other things. What would stop you from building, in this way, a complex assembloid with, maybe 10 spheroids? Ultimately, it's going to be limited by the lack of vasculature and the inability of diffusive transport processes to sustain life at the center, of one of these more complex assembloids. And so what you'd like to, introduce into this context is some mechanism for introducing artificial vasculature that could enhance transport of nutrients in and waste out, to maintain viability of these larger, artificial tissue constructs. And the question is, can you create 3D microfluidic networks that resemble those that are observed in biology? And so this is a glomerulus. It's a filtration unit that occurs in the kidney, and then this is a decellularized brain showing vasculature there. So it's amazing level of hierarchy in the design and full 3D volumetric penetration. And the same ideas actually work quite nicely with microfluidics. And so you can create now 3D microfluidic constructs that resemble those that occur in biology, and you can pair them with electronic and optoelectronic functionality to sustain larger artificial tissue constructs. And I won't go through all the details, just spend a lot of time on this. It'll be published in Science Advances soon. It's been accepted. It's in press there. But now you can move microfluidic structures from 2D to 3D, and you can create structures for interconnecting and addressing multiple 3D microfluidic structures through the base elastomer that you're using to drive the assembly, which is what's being illustrated here. And you can do multi-layer sort of space filling, same type of concepts I was describing in the context of electronics, but now microfluidics with geometries that can be as small or even smaller than those of capillaries, the smallest capillaries. So with cross-sectional dimensions in the range of an individual blood cell, so you can go all the way down to the smallest dimensions needed for artificial vasculature. There's no limitation there, and you create all kinds of large areas, sort of hierarchical type of the constructs is just being illustrated here. It's all guided by computational mechanics modeling, and then we can build these structures in the lab as well. Here's another level of 3-dimensionality. You've been thinking about, like, how you build a glomerulus, and this is the way you do it. So this is a little bit more advanced than what I was talking about before. It's like 2-level of 3-dimensional transformation. It's like planar to 3D and then to full 3D, where the entire substrate actually morphs into a sphere. In this case, just kind of a engineering challenge we got interested in and thinking about that. But you can pump blood, artificial blood, into these systems, and you can integrate them with living tissues and sustain life throughout 3D volumes in ways that would be impossible without the vasculature present. And I won't get into all the details, but naturally, you can combine the optoelectronics and the electronics with the fluidics. It's very easy to do that. So now you have kind of an integrated microfluidic electronic network, and that works without much problem. You can do that as well. And so this is just kind of a fanciful, modeling demonstration of the kind of versatility I think that's present with these kinds of geometry transformation methods, and they work with all kinds of different materials. I've made that point a couple of times already, and anything you can do in a planar geometry, you can down do in 3D is the idea. And I think that creates some opportunities in biointegrations, kind of where our focus is, but maybe other areas of technology as well that you might want to think about. So I feel like I'm out of time, maybe even a little bit over. So I'm going to go ahead and conclude. Everything that we do is highly collaborative. We couldn't do it without really outstanding senior collaborators. I've mentioned Yonggang Huang, Yihui Zhang at Tsinghua, very, very important theory collaborator, Ralph Nuzzo, John Sweedler, Colin Franz, John Finan, others on organoids and studies of neurons. But I always conclude with, acknowledgement of the students. I've been very blessed with incredible students and postdocs. Shinga is on the line now. Really great postdocs who came through the group and running their own independent research programs, doing extremely well. So I like to acknowledge the people who actually do the work. I just talk about it. So I'll conclude by acknowledging them and thanking you again, Paul and Alice, for putting together this event. I'd be happy to answer questions if there are any.
Thank you so much. There are indeed questions. And then also, please stay tuned for our panel to come. I think I can put this back up. Great. So the first question is from Ximing in Shenzhen. And he says, Professor Rogers, thanks for your wonderful talk. As you demonstrated, the 1,000+ channel devices for chronic recording, comparing with the complexity of neural systems, is really small. You mentioned, but he would like you to comment further on how you'd like to improve the device, including scale up to the big arrays that you mentioned, or further miniaturization to higher resolution.
Yeah, so thank you for those questions. So how do you scale up and how do you scale down, I guess, in terms of resolution and size? And I guess we've thought a lot about it. I mentioned, this paper that we published a year or two ago around, an approach that would align with foundry level manufacturing capabilities. I think you probably need to do that. I think without that, it's going to be a really hard slog, to get to like 1 million channels or 100 million. You want to be able to use, technologies that are TSMC or, wherever, you'd like to be able to use nanometer scale, transistors and leverage all of that. So I think that's probably the way to go on scale up, is to, tap into that foundry level manufacturing capability for the integrated circuit components. But ultimately, you need to distribute that functionality, and you don't need it at full area coverage. You couldn't really even use it at full area because you wouldn't have the mechanics you would need. So you need very tiny circuit blocks, I guess, at each node in the array. And you need to be on a flexible support, obviously. So I think, ideas that Paul and I have been looking at in transfer printing, many other people too, doing great work, but, somehow uniting, soft, flexible, biocompatible materials with state-of-the-art silicon CMOS. That's kind of probably what you need to do. It seems like it'd be hard if you don't do things in that way. So it's got to be some kind of scheme like that. And we have our own ideas, but I also appreciate a lot of creative people out there. Maybe they have better ideas. I don't know, but that would be the vision, like a hybrid electronic system. I'm pretty sure that's what it's going to need to look like. And we feel like, those approaches scale pretty nicely. Now the details are like incredibly daunting. So I wouldn't want to like sweep those under the rug. I mentioned, like data management, power management, thermal load, like if you think about like a thermal budget associated with the brain, it's like incredibly narrow. Like if you're heating up brain tissue by more than a couple of degrees, it's game over. So, and you can thermally modulate all kinds of stuff that, you know, thermal management in a piece of consumer electronics is one thing, but you think about the budget in the window, you have to operate over, in a biological context, it's like way more challenging. So like, how do you do ultra low power, and how do you do local signal aggregate? Maybe just count spikes. You don't need the full, electrical waveform associated with neural activity. You just need, was there a spike or not? How do you downsample and things like, millions of questions like those. But like conceptually, like from a materials standpoint, I think it probably would play out in the way that I described. So that's kind of a couple of thoughts. That the other the couple of things, how do you do stretchable? I think that's really hard, and it comes down to the question of how do you encapsulate these devices? Let's say you want to do a deep brain stimulator, or an equivalent of that, maybe a surface mounted stimulator to address epilepsy. You'd like to develop a device in that context that would last the life of the patient. So let's say that's 10 years. And then let's say you probably want to over-engineer it. So you would like to have it last 100 years. So how do you develop a piece of flexible electronics that can operate in a stable fashion for a century in warm salt water? That's pretty hard, and how do you do that in a very thin form factor? You can't just put it in a ceramic case, like how do you do that? So we spent about a half a decade worrying about like encapsulation layers and like how to solve that challenge. It's not very sexy in a sense, but like if you don't solve that, you don't really have a realistic system. So there are lots of details like those, but that's maybe a few thoughts. The other question was around how do you make the devices smaller, maybe improve the resolution, and there I think maybe it's a little bit simpler. You probably don't need resolution beyond maybe a couple of tens of microns. Like having subcellular resolution might not be needed or valuable. You're looking for networking level activity, I think. So getting to like kind of a 20, 50 micron pitch is straight, that can be done. I mean, I think you hit the resolution requirements pretty quickly. I don't feel that that's a challenge. Probably the bigger challenge is around the physics of how you pick up electrical activity from individual cells when you're also measuring the effects of other cells that are in adjacent to or in proximity to the actual node that you're wishing to sample. So there may be some more fundamental aspects of how crosstalk happens, I guess, but from the engineering standpoint, you can get to the resolution that you need pretty easily. I think scaling the area and scaling the channel count's probably more challenging than hitting the resolution requirement.
Mm-hmm. Actually, the power question leads right into the next query we have from the audience. Let's see, that's from Hua at NUST. Professor Rogers, great talk. Thanks and congratulations for implantable devices, batteries, and data transmission always generate heat. How will that affect the animal's behavior and or damaged tissues?
Yeah, great question. So I mentioned a couple of things about thermal load. So, so a couple of like thermal management issues, and I talked about a lot of several different systems anyway. I mean, maybe just to separate the conversation, you have like thermal associated with the power harvesting supply unit and the wireless data transmission components. That's one. Those are typically a little bit easier to manage because you can move that function away from the neural tissue, put it subdermally. You have a little bit greater budget there, and then you can exploit different strategies in heat spreading and different things, and you can sort of, I guess, separate the engineering challenges associated with those components of the system from those associated with the actual neural interface, whether that's an electrical interface or an optical interface. That becomes a little bit more challenging. You have a little bit of I would say far fewer options in how to manage that. So for electrical sensing, it's typically not a problem. I mean, it depends on the design, but there's not a lot of power consumption locally at the sensing node. Simulation may be a little bit different. It would depend on the details. We've spent probably and we do IR imaging, which is just to check there isn't any significant heating in those 1,000 channel systems. It's really all of the power is being dissipated in the wireless communication and in the back end, I guess, separated from the brain itself. The bigger challenges probably are around the optical, because the LEDs have some finite efficiency. I mean, even you're using very advanced indium gallium nitride LEDs. The amount of power that you supply to the LEDs that ends up as a heat source is considerable. Maybe it's 80%, 70% of the power you put in ends up as heat, and you have these tiny LEDs, and you think about like power management, like in an LED flashlight. I mean, you're used to seeing like giant heat sinks and stuff, and now you have one of those types of light sources embedded in the brain. It seems like that would be highly problematic. And we spent a lot of time on that question before putting any of these devices in animals, using phantoms and modeling and other things. And so I guess what saves you is that for optogenetic stimulation, you're typically operating in a very low duty cycle mode. And so the LEDs are not on for very long periods of time. And so in between the times that they're on, you just have passive thermal diffusion, sort of reducing the peak temperature at the device location. That's one. The other thing is that the devices themselves are very small, so the surface area to volume ratios are very high. So you end up with very high rates of passive thermal diffusion as a result of the small sizes of the devices. And then the other thing is there's a lot of flow of fluids through the brain, not just blood flow, but also flow of cerebrospinal fluid. And that's almost acting like a liquid heat sink, to dissipate heat as well. You put those three sort of factors together, you end up with peak temperatures that are very low, maybe a factor of 10 below any kind of threshold that would lead to thermal induced modulation or ultimately damage to the tissue. So that's kind of how you end up managing thermal for these optical interfaces. But it's a super important question. I think Xing maybe has done some work on integrating nanocrystalline diamond materials into the probes to sort of facilitate thermal transport along the length of the probe away from the light source. That's a really great idea. So again, some materials approaches that might be important. The other thing is you can embed sort of transparent optical materials on top and below the LEDs without affecting the optical coupling very much, but they come thermal isolation layers. You can do that as well. We haven't had to do that for our devices, but you can do that. So I mean, maybe an affiliated point here is that we got involved in this area, published our first paper in 2013. We got a tremendous amount of interest from the neuroscience community. So there is a small startup company that we put together in 2015, 2016 to distribute the technology to the neuroscience community. I think we've sold 250 systems or so globally. So it's probably representing now the standard technology for optogenetic studies across probably a third of the neuroscience research community, something like that. So these are used very, very widely at this point, these penetrating probes with the tiny LEDs, and seems to be working fine. But you do have to work out all of the details. These are great questions. You can't ignore some of these things. Otherwise, you end up with all kinds of artifacts that corrupt your neuroscience studies and make it more difficult to get any kind of real insights into the biology.
Terrific. We have one more question from the field and then another late one that came in from a colleague here. The next question is, Dear Professor Rogers, thank you so much for your talk. The 3D structures from your group are amazing. Do you think that it is possible to get liquid metal into your 3D ribbons to drive structural deformations?
Yeah, that's a great question. That's something we're working on right now. So that was a very insightful question too, I guess. So we've been doing that now for a while, and you can use the liquid metal in a few different ways. One is you can exploit the phase transition, the temperature induced transition, liquid solid, to sort of reprogram shapes and mechanical properties in these 3D networks. That's one, but maybe more interesting. I don't know if this is what you were suggesting, but you can flow current through those liquid metal wires, I guess, embedded in these channels, and cause them to interact with externally applied magnetic fields to create Lorentz forces that can reconfigure the shapes of these 3D structures. So it's kind of a little bit exploratory, just thinking about what's possible. But those appear to be viable options. If you're interested in shape change, like programmable shape change, that's one way that you could do it. So liquid metals are a good match for these kinds of frameworks.
Mhm. And then our final question before we go on to the panel comes from our colleague at UCLA, Professor Zhangfeng Duan, who says, thanks for your very inspiring talk. Is cellular level conformability and permeability an issue to consider when constructing electronic tissue interfaces?
Sometimes, yeah, good to hear from you, Feng. So I would say sometimes, not always though, because if you think about the LEDs, they're spewing photons out in all different directions, and you're not controlling where those photons are going. So cellular scale, like smaller, is probably better just in terms of mechanical and volumetric load, from the standpoint of the device. But it's not as though cellular scale allows you to illuminate just a single cell. I mean, you have an illumination volume that is about a 0.5 millimeter in diameter. If you think about the absorption and scattering, and you do Monte Carlo simulations, that's kind of what you end up with. So you're illuminating that whole volume. The cellular level specificity is occurring from the standpoint of the genetics. So the viral vectors are designed to infect only certain cell types, and then the genetic code that's injected by the virus causes the expression of these light sensitive ion channels. And so you're illuminating the whole volume, but only those cells that have been genetically modified are responding to that light. So we like to think about cellular scale, and you can actually go, like I said, much smaller than cellular scale. But at least for this kind of application, is probably diminishing returns. Once you're kind of at a 100-micron scale, going smaller is not buying you much. I guess maybe from a thermal standpoint it's helping, but from the standpoint of targeted illumination of tissue, at least the way that we have things set up, it doesn't really affect too much. You're really just limited by photon diffusion and absorption.
Well, thank you very much. Let me take the screen over here for our staff in Beijing. And then I will introduce our panel. There we are. So our first panelist is Dr. Xing Yu, who's an assistant professor of biomedical engineering at CityU Hong Kong. Professor Yu's research group focuses on skin integrated electronics and systems for biomedical applications. Dr. Yu serves as associate director of the Chinese Academy of Sciences CityU Joint Lab on Robotics, and is an associate editor or editorial board member for 5 journals. He was named a top 35 innovator under 35 in China by MIT Technology Review, a new innovator of IEEE Nanomed, and won the MINE Young Scientist Award. Our next panelist, Dr. Xing Shen, is an associate professor in electronic engineering at Tsinghua University. He explores implantable, micro and nanoscale optoelectronic devices to enable high performance and versatile applications in biomedicine. He's an associate editor for OSA Optical Materials Express. He's the recipient of Young Scientist Awards in Progress in Electromagnetics Research Symposium Peers, and also Microsystems and Nanoengineering Summit MINE. And our challenger today is Wei Ling, who's working towards a PhD degree in biomedical engineering at Tianjin University with Professor Shan Huang, and has won the National Scholarship several times and the championship of the first ICANX Academic League in 2020. So let's bring everybody out. Hopefully you have your cameras on. And let's start with Wei Ling.
Wei, are you ready, please?
Yeah.
Yes, we'll give you the first two questions.
Yeah. Thank you, Paul, and thank Professor Rogers for your excellent talk. Your talk is always impressive, and I'm interested in the 3D mesoscale structures. As you mentioned, these structures can encode the organoids like a cage and measure multiple biological information. So I'm curious about whether these devices will have some adverse effects on the growing organoids and how to remove these defects. Thank you.
Yeah, that's a great question. So the way that we've used these interfaces is in their application to organoids that have already matured. So they've already grown to their kind of targeted size, and so they're not continuing to grow once we insert them into these baskets. But it's a great question because I think we'd like to be able to follow the growth pattern, right? And maybe probe these things as they're growing to their matured state. And I guess there are a couple of ways that you could think about doing that. And we haven't done it yet, but I think it's a great question and something we've been thinking about also. One is, I mentioned that the way that these 3D structures are formed are based on an elastic buckling mechanics that does the transformation. And so these frameworks are still sitting on the elastomer. So as I mentioned, you can kind of open them up to put the organoids in. So you can imagine sort of a stage that would adjust the strain in the substrate to allow the basket to gradually increase in size, right, over time to kind of follow the growth dynamics of the organoid, and that would be really interesting. We have done a lot of mechanics modeling of the forces that are induced, at the interface between these baskets and the organoids. So we know pretty well what those forces are, and you can maintain them at a very low level. In fact, the electrodes don't have to be directly in physical contact. As long as they're close, within maybe 10 microns or so, the surrounding solution is ionically conductive anyway. So you're coupled in through the surrounding cell culture media to the organoid itself. So to your other question, do these baskets have any adverse effects on the organoids? We haven't seen anything. And in fact, you can keep the baskets out of direct physical contact to further reduce the possibility that they would have any kind of impact on the natural growth pattern. So those are my two answers. You could follow the growth by manipulating the strain in the substrate, thereby changing the geometry of the basket. And furthermore, if you have appropriate control systems, you could maintain a small proximity separation between the devices in the basket and the organoid itself to eliminate any kind of physical impact on how the organoid is behaving. But those are good questions, and we've thought about them. We don't have the growth experiments directly, other than that neurite bridge. But there, the overall geometry is not changing much. It's just like growing across that gap. But thanks for those questions.
Yeah, thank you for your answer. And I have another question. After we have obtained the abundant data using these novel neural interfaces, so how can we use this data to help us better understand the working mechanism of our brain?
Yeah, so that's another good question. I mean, you have to be careful because, at least for the organoids, because there are tremendously sophisticated optical techniques, all kinds of tagging approaches, genetically encoded fluorescence indicators, not only for calcium, but for various neurotransmitters, even transmembrane potentials can be determined. And so for these kind of hardware materials oriented approaches, you have to think very carefully about what kind of measurement capability are you going to provide that is not already possible with sophisticated optical techniques. That's the competition, I would say. And there, global communities developing new, genetic encoded, fluorescence indicators and so on. And so I think, direct, and you can do a lot optically. So I think anything that involves mechanics, probably, you're not going to be able to do that as well optically. I could be wrong about that, but just thinking kind of out loud in response to your question. So we have the ability to put strain gauges in these devices. We have interfaces with cardiac organoids, for example, so you can measure stresses and strains associated with beat cycles. We have devices that they're using hybrid sort of biobots, where you have muscle tissues as actuators, and you can measure all sorts of things that are associated with the mechanics, tissue mechanics. We have paper and advanced materials using these structures as basically frameworks to hold them so we can evaluate mechanics. So anything along those lines. I think direct electrical stimulation, electrical monitoring, probably benefits from a physical interface beyond just an optical one. Maybe thermal modulation, you need some kind of actuator there, but maybe you could do that optically also. Anyway, you have to think pretty carefully so that you don't end up spending a lot of time developing a technology that is easily replicated or maybe outpaced by some optical scheme. You don't want to do that. You end up wasting your time. So yeah, I have to think about it pretty carefully. But I think so some of the demonstrations that we've done, I think, fit into that kind of area of uniqueness where you need a physical materials level interface, but complementary to other schemes for monitoring. That's in the context of like in vitro studies, and you're looking at organoids. I think optical techniques are probably, like imaging techniques, are probably much less applicable to moving animals or humans, for example, just because of the artifacts, the bulk, and the power requirements, all of the difficulties associated with doing optical imaging, in an ambulatory living organism. So there, I think there are more compelling opportunities for a hardware solution. And there are sort of head-mounted 2-photon imaging systems. You've seen them maybe used on mice. I mean, it's crazy. Like the microscopes and the head mount is bigger than the animal. It's like this gigantic thing, and so you probably use it in certain instances, but I think it's probably not ideal.
Thank you very much.
Terrific. Thank you. Let's move on to Professor Shang. We'll work across the screen here.
Oh, hi, John. Great work as always. Great talk. Yeah, I may have a general question. As I learned from your lab, right? In the past 10 to 20 years, you have been spending most of the time to design and engineering the electronic and optical materials and devices to form ideal biointerfaces, and you actually become a pioneer in this kind of area. And I actually noticed that most of your works actually focus on the optimization, the development of these material and devices in terms of their geometry, the structure, and the mechanical properties, and to optimize these properties and fit with the biological system. And what are other considerations in terms of, for example, the chemical properties and the biological properties for these material and the devices, and make them?
I think it's a great question. I wouldn't argue with your characterization. I don't like to hear 20 years. It makes me feel really old, but let's say 10 years. Make it a little bit shorter, but that's probably an accurate characterization. We kind of focus on geometry, mechanics, form factor, I guess, biocompatibility at that level. I think we've sort of emphasized those aspects because I felt like when we got started in this area, it's probably an underdeveloped aspect of implantable body integrated devices, the mechanics, geometry, form. Whereas, studies at the materials level in terms of biocompatibility is much longer history, and incredibly deep, base of literature already, on those types of topics. And so we felt like there were lower hanging fruit where we could bring, some kind of contribution to the community by focusing on these other things and kind of leveraging the existing base of knowledge around materials level biocompatibility. That's not to say that there aren't opportunities in developing fundamentally new materials where the focus is on, the chemistry of biocompatibility. I think that's a great area for research. We just decided, maybe we could make a contribution more effectively by focusing on these other things. I'm not a synthetic chemist, and so kind of limited also just in terms of my own expertise, I would say. But we have spent a lot of time in recent years around hydrogels and thinking about hydrogels as a coupling medium. So we have all these stretchable, flexible platforms, all the functional nanomaterials, and ultimately like what is the material that's establishing the direct interface to biology. And I think David Mooney and Jigang Suo and Xuan Zhao and a number of others have been kind of working on hydrogels. I think not so much in the context of bioelectronic devices, and so we've kind of jumped in and been able to kind of build on the chemistries that they've put together over the last few years and extend them to allow for a coupling, kind of bidirectional coupling biological tissues on one side and our style of bioelectronic devices on the other. So kind of like tailored to enable adhesion on both those surfaces, and still thinking about the mechanics, but I guess the efforts are a little bit more centrally focused on the polymer science and the materials of the hydrogels. We have a paper coming out in Nature Materials on our work. It's like a quite comprehensive study, but so I would say we're kind of doing things in that realm at this point. The other thing I would say is that I didn't talk about it, but I mentioned at the beginning of this talk, the idea of bioresorbable electronics. That's all materials. I mean, it's like, we're building on ideas in flexible, but it's all about new polymers and metals and what are the routes for chemical degradation and hydrolysis and enzymatic decomposition, and that's a whole separate centroid of activity, as in the group. And I didn't touch on it during this presentation, but that's where our kind of base materials level work is really focused. I mean, material is very important, what I talked about here, but I would say mechanics and integration and manipulate, that kind of thing, is a little bit more central to that aspect of the work. So anyway, a little bit of rambling answer to your question, but those are a couple thoughts.
Okay, thank you. And my second question is more technically related, and very interested in the last slides about the most recent work on the artificial 3D vessel structures. And have you considered how do you engineer such a structure to interact and, for example, exchange chemical signals with the cells growing on inside? How do you?
So there are a couple of things. A couple of things. I mean, you need to engineer permeability in the walls of those microfluidic systems, and there are a couple of ways to do that. I mean, the silicone PDMS materials we're using currently have some level of permeability themselves, and so you can just make, say, the top surface wall of those microfluidic structures thin enough that you just get diffusive transport right through the silicone. So you can do that. The other thing that you can do is you can lithographically define porosity, or you can introduce porosity using sacrificial granular material when you're casting the PDMS, and then you dissolve it out. So you end up with transport pathways through the voids that form as a result of dissolution of those grains. So there are different ways to do it: deterministically introducing porosity, kind of at a more of a materials level, or just exploiting the intrinsic permeability of the material. So you can do that. I didn't embed the results of those kinds of studies in this talk, but we've looked at that extensively, because as you're suggesting, the whole point of the vasculature is to allow for a transport pathway for nutrients in and waste out. And if you don't have some kind of permeability pathway to the fluid component of those microfluidic structures, it's going to be ineffective. So there are a variety of ways to do that. And I guess the other thing is, if you're doing it lithographically, you can define the spatial distribution of where that diffusion occurs just by defining the geometry of the pore structures. So there's a tremendous amount of flexibility in how you design that.
Okay, great, thank you, and looking forward to your new results.
Yeah, thanks.
Thank you very much. Let's move on to Professor Yu.
Thank you, Professor Weiss. Thank you, Professor Jiang. Georges. Yeah. It's a really nice talk. So always. So brought my memory back to 2015 because I still remember the first project you assigned me is a 3D work. So it's assembled these 3D PCT on their 3D structures. And so 6-7 years passed. It's really amazing see the progress of the 3D works, and especially when I saw the result of the 3D microfluidic work. Is really amazing. And I have a question about the 3D works. So can you talk about the future opportunity to combine the 3D structure, especially the compressive buckling with soft robotics?
Oh, that's an interesting thought. I've always felt like there are probably really strong synergies between kind of the soft robotics community and the soft electronics, or maybe you're suggesting microfluidics as well, communities. There's not as much work at that interface as I would have expected. We're kind of maxed out and we're not, able to devote bandwidth to soft robots. But I think it's a really interesting field of research, and for people who are working in flexible electronics or flexible like 3D microfluidics, there's probably a lot of really great areas and directions to go. I guess, in real time responding to your question, I guess, that this question around liquid metals came up, and we are doing Lorentz force actuation in these microfluidics. Is probably not what you're suggesting with your question. I think you're probably thinking about pneumatics or hydraulics as a actuation mechanism, but Lorentz force is another way to think about artificial muscles, and it has great synergies with these kinds of 3D microfluidic frameworks when filled with liquid metals that can provide a electrical conduction pathway, but also don't constrain the mechanical properties because it's a liquid. So even relatively modest forces that you can generate using Lorentz phenomena can be sufficient to move these structures around. I don't know if that's a realistic basis for a robot, but it is a realistic basis for a programmable 3D framework. You can move it around. The forces are it's maybe not sufficient as a muscle, that would cause a robot to walk or grasp an object. So maybe you need some kind of mechanism that offers higher force density, and maybe you could use pneumatics. I guess we haven't thought about that explicitly, but I think it's an interesting idea. Maybe you can come up with some schemes and do something in that area. I do remember you're a key person in some of that early work in 3D PCT. I think it was published in Nature Electronics, if I remember right, with Mungdi, actually, former graduate student of Alice. So small world, I guess.
Yes, Magni is here.
Yeah.
That actually brings me to a last question, and that is, you span so many different fields, and it was clear from your answer to the last question, you choose to move forward in some and not approach others. How do you gather information across all those fields and then decide, here's where we can either solve, fulfill a need or move the state of the art forward, and here's where we don't want to put in effort?
Yeah, that's a great question, Paul. I guess it's a little bit of a gut instinct. At some level, you have to realize that you're good at some things and maybe not that good at other things, and you want to kind of gravitate toward projects that have strong synergies with your own expertise and your core competencies. So that's probably one of the most important considerations. The others are, do we want to work in a hot, sort of trendy, crowded field? Probably not, just because, I think we're productive, but we're not fast. That's not our core, skill in being able to, win foot race against some other group, in a heavily populated space. And so we try to shy away a little bit from those kind of areas. Not that, one shouldn't do research in those types of fields, but, perovskites or graphene or 2D materials, it's great areas for research, but I don't think we could really compete effectively because we're probably not like just fast enough. It's a little bit slow, I would say, at some level, but maybe deliberate. But and then the other aspect of that is that if you're working in a crowded space with a long history, then you have to really study the literature and think about whether your idea has already been tried out before. It's just harder, I guess, to contribute or come up with really impactful ideas in a field that's heavily populated with a long history. And so per my answer to Xing, I think, at materials level biocompatibility, that's a, a field with a very long, rich history. And, are we going to be able to do something in that space that hasn't been thought of before? Maybe, but maybe not. And so if we have other directions that we could go, a little bit less populated, maybe a little bit more off the beaten track, then we would prefer to do that. The other two considerations I would say is, everything that we do is, well, not everything, but I would say most of the best stuff that we do is highly collaborative. So is there a great collaborator, that is interested in working with us, and are they leaders in their field, and are they collaborative type? That's an important consideration as well. Go in a new direction. You'd like to be able to leverage your skill set against some other high level person. Well, a high level person, let's say, in some other field that's complementary to your own. That would be important. But I would say, as you get older, at least as I get older, you'd like to be working on a topic that has the potential, at least, for real impact. And I think a lot of people kind of think about that, but as you get older, you think more about that. Like, what am I doing now that could make a dent, at a broader level? And for us, a lot of that is human health related. Like, are we working on something that has the chance for impacting patient outcomes, that type of thing? That's a pretty significant consideration. It could be near term, it could be a little bit further out, but like, if what we're doing is successful beyond our imagination, like who would care about that, and who would it impact in terms of what we're doing in biomedical engineering. So anyway, those are kind of some of the considerations, but it's not like we have a checklist. Like I said, it's more of a kind of a overall gut feel where you kind of like somehow integrating these different considerations in some way, and then making a decision. The final thing I would say is like student interest. If a student's not interested in it, you can't do it. Like, even if you wanted to do it, you can't like force that. I think student enthusiasm and passion for different projects, ultimately, I would say that's having a huge effect on like the progression of what we're doing at a research level. You think you're guiding the process, but I think at some level the students are choosing what you do, either directly or indirectly. The most passionate, productive students are producing the most science, and then that's leading the largest grant opportunities, and pretty soon your your program is sort of shifting in that direction. So that's an important consideration, and like an uncontrolled parameter, I guess, at some level, is this like the student interest.
Indeed. Well, thank you so much for not only coming back in the presentation, but this incredibly stimulating discussion. Of course, we wish we could do this in person, and that we could walk across a stage and hand you a certificate. And we're looking forward to sometime very soon when we can get to see each other in the world. In the meantime, we've been able to pull people from all over the world together this evening, afternoon, or morning, depending where one's located. So, thank you once again, John, for just a spectacular tour through a part of your science, engineering, and laboratory.
Yeah, thank you, Paul, for hosting and the intro and managing the Q&A here. And thanks, Alice, again for the invitation. It's a pleasure to have a chance to participate in this seminar series.
Great. And coming up next week, we'll have Professor Masayoshi Isashi, hosted by Alice, and more to come and the rest of this month. And I think we almost have our schedule complete through the end of the year. So we'll look forward to seeing you again soon on a Friday. And now please enjoy your weekend and come back. Thank you.